Hello, everyone, and welcome to today's webinar, Constructing a Spatially Resolved Single-Cell Atlas of the Mouse Retina with the MERSCOPE platform. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by VizGen. To learn more, visit vizgen.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Dr. Rui Chen, Director, ATAC Single-Cell Genomics Core, Baylor College of Medicine, and Professor, HGSC, Departments of Molecular and Human Genetics, Baylor College of Medicine. Dr. Chen, you may now begin your presentation.
Hello, everyone. Thank you very much for the introduction. It's really my great pleasure to be here to share with you what we're doing here at VizGen. So for today's presentation, I hope to first give a quick overview of our platform and illustrate how you can apply this technology to really gather new biological insights and map the future with spatial genomics. So as many of us know, biological tissues are very complex and made of many different cell types that express genes very differently. And understanding that gene expression profile within each cell and across the tissue is very critical for better understanding of the biological system. So over the past 2 or 3 decades, we have seen a revolution in genomics research, which really enables us to characterize different biological systems at unprecedented scale and resolution. As you can see from this landscape of genomic evolution shown here, from bulk RNA sequencing to single-cell sequencing, we can now interrogate the gene expression profile of individual cells and really use this information to understand cell composition and cell-cell interactions now. However, sequencing-based approaches have a fundamental limitation because they don't usually provide the spatial context of what you're looking at. To do the sequencing, cells either have to be destroyed or dissociated, and thus we lose the very rich spatial information for the 3D tissue structure. And as many biological problems do require multiplexed, spatially resolved single-cell measurements, we see a huge demand for technologies that can satisfy this criteria, and that's what we are working on within VizGen. So specifically, we are commercializing a technology called MERFISH that enables in situ single-cell transcriptome profiling, and our commercial product, MERSCOPE, pictured here, is the complete platform solution for MERFISH technology. So MERFISH stands for Multiplexed Error-Robust Fluorescence in situ hybridization. So this is an RNA imaging-based technique that can locate and quantify hundreds to thousands of different RNA species at the same time with subcellular resolution. So as illustrated on this slide, there are 3 key aspects that makes this highly multiplexed approach possible, which are combinatory labeling, sequential imaging, and error-robust barcoding. So to perform MERFISH, we use a tile of oligo pools which contains different barcodes to label the different RNA species. Then each barcode is fluorescently detected in sequential round of imaging, and the optical barcode generated from these many rounds of imaging with 1 indicating fluorescent on and 0 indicating fluorescent off, then this optical barcode will help to resolve different RNA species. And in the end, it will generate a spatially resolved RNA profiling data, and in the end, it will give a physical picture for the cell or tissue of interest, as illustrated by the picture on the right, with colored dots indicating different RNA species and gray lines indicating the cell boundary. So MERFISH was invented by our scientific co-founder, Dr. Xiaowei Zhuang at Harvard University back in 2015, and since the initial publication in the Journal of Science, it has been validated by a long list of publications and brought many transformative discoveries already. And actually, spatially resolved transcriptomics was selected as Method of the Year in 2020 by Nature Methods, and MERFISH was featured there, really highlighting the excitement it has already generated. So to make this powerful technology more broadly available, we are commercializing MERFISH through our commercial platform called MERSCOPE, and this platform facilitates the whole MERFISH measurement with a complete reagent, instrument, and software solution. Upstream, we have the ready-to-use MERFISH gene panel and sample prep reagents to stain the samples. Then the processed samples will be loaded onto the turnkey instrument for imaging, and we also have software tools to facilitate the data analysis and visualization. So as a targeted RNA imaging technology, first you need to stain the samples with the gene panel, and then the tissue and slices then will be loaded to the MERSCOPE flow chamber and put together. There is the associated reagent pack that goes with the sample. Then it will be loaded to the MERSCOPE machine. And as illustrated on this slide, MERSCOPE has very large imageable area, up to 1 square centimeter, with imaging resolution down to 100 nanometers. So with that, the instrument will automatically acquire the data and transfer the data to. With that, the instrument will automatically acquire the data and then transfer the data to the imaging processing computer for analysis, as illustrated by the Wisdom data flow for MERFISH here on this slide. So there are 5 different data outputs coming out of MERSCOPE analysis. The first one is the list of all detected transcripts, which contains the gene counts and then the X, Y, Z coordinates. The second one is the mosaic images, which contains the DAPI and total mRNA staining, as well as some other protein staining images in the TIFF format. Then the third one, transcripts per cell matrix, is a format very similar to what single-cell RNA-seq data output looks like, which contains the gene and cell matrix. And finally, we do provide cell metadata that contains the X, Y coordinates of the cell, as well as the polygon shape of the cell boundaries in HGSC format. So you can use software package such as ScanPy or Seurat to analyze the data. And alternatively, we have also developed MERSCOPE Visualizer to facilitate the data analysis and visualization, as well as illustrated by this slide here. So as a gene expression profiling tool, MERSCOPE at its basic form just reports the location and quantities of RNA species, as well as the cell boundary information. But with so much information, it opens up many application areas, and we envision it can be broadly used in many research areas, as shown on this slide. For example, you can use this to profile the gene expression in situ, classify and discover cell types and states, and reveal the cellular structure and organization within native tissue context, or even profile the cellular activity. For example, shown on this slide, this is using a panel of around 500 genes to profile the mouse GPCR and RTK receptors in the mouse brain, and you can see that this is a highly quantitative and highly reproducible measurement. From the left, you're seeing the 8 RNA subset out of this 483 measurement, while in the middle, you're seeing the gene expression within single cells. So this has also been used in more clinically relevant samples. For example, showing here is using a panel of 417 genes in human colon cancer. And again, which is only 10 RNA subsets selected out of these 417 genes. There is already a lot of biological insights that you can gather from such a single measurement. So within Wisdom, we keep pushing the limit of the technology by expanding the application areas. We have proven MERFISH in more than 20 different mouse and human tissues, and a few representative tissue types are shown on this slide. And I hope that these beautiful images illustrate how incredible spatial architecture of different tissue types have, and how important it is to have this highly sensitive and versatile tool to profile the gene expression and cell types across different tissue types. And finally, just to add on, so MERFISH is compatible on many different sample types, including adherent cells, suspended cells, fresh and fixed frozen tissue blocks as well. And last, just to recap what I have just shared, here is a really quick summary of some of the MERSCOPE advantages. First, MERSCOPE has very high multiplexing power. We can measure up to a few hundreds to thousands of genes simultaneously in a single sample. And secondly, MERSCOPE has very high spatial resolution. We can localize RNA transcripts with subcellular resolution, and that high spatial resolution is preserved across the entire tissue. Thirdly, MERFISH has very high sensitivity for identifying RNA in situ, and it really enables us to detect many lowly expressed but functionally important genes very easily. Furthermore, MERSCOPE has very high cell throughput. We can profile square centimeter of tissue with hundreds of thousands of cells and sometimes millions of cells in a single experiment. And importantly, no follow-up sequencing is needed. You get all the information in a single experiment run without any additional sequencing cost. And finally, MERSCOPE is very flexible. We can perform MERFISH of almost any tissue or sample types. So in summary, I hope this quick overview, this gives you a quick overview of our platform, and we're really excited about the potential of our technology, and would love to hear how we could potentially help with our help your research with our platform. And with that, I wanted to turn to Dr. Chen, who will be sort of sharing his insights in using MERSCOPE and image the retinal tissue. Thank you.
So first, I'd like to thank for the introduction, and also really happy to have the opportunity to share with you about our experience with the MERSCOPE platform, using this wonderful technology to build single-cell atlas of the mouse retina. So our lab is generally interested in the visual system, in particularly the retina, neural retina, which is at the back of the eye. This is part of the organ that capture the light and convert the light to electric signal and then transmit to the eye, to the brain. So this part of the neuroretinal tissue is actually the leading target for the irreversible blinded diseases. So although it's only have 5 major neuron types in the retina, the number of cell subtypes based on the morphology and the physiology is quite complex. So as has been estimated, about 60 to 70 different subtypes exist in the human retina. And add on to that complexity, the cell proportion of different subtypes are quite different. For example, in ganglion cells, which make up about 1% of the cell population, there's about 15 different types. Quite interestingly, the mouse retina seems to be even more complex than that in humans. So it has been shown based on the transcriptome and other studies, there's over 100 different subtypes identified in the mouse retina. And this is a UMAP based on single-cell RNA-seq of about 250,000 mouse retina cells. And we can see all the major cell types can be identified. There's more than 100 subtypes. So this single-cell technology is really wonderful that so now for the first time we can identify all the subtypes. However, what's missing is the spatial information. Just like if we think about the retina as like a really nicely arranged fruit tray, what we have right now instead of smoothie, which we getting from bulk sequencing, from the single cell we can find all the cell types and how many of them are there, the proportion. However, what's missing is the spatial information, which is very critical for us to understand the function of the retina. So there's basically rapid progression in the single-cell spatial transcriptome technologies that can be used to get to actually obtain that spatial information at single-cell resolution. Simply put, there are 2 major types. One is the sequence-based type method, one is hybridization-based. For the sequence-based, for example, from the Visium technology from 10x is something we are quite familiar with. And today, I will mainly focus on the hybridization-based method, particularly MERFISH here. So Dr. Jiang already went through the background how the MERFISH work, so I will skip this slide. So we are actually lucky to be selected as one of the early access sites for the MERSCOPE. So instead of the nice-looking box you see Dr. Jiang presented, what we have is this basically the gut of the instrument. There are 2 major parts. One is the imaging part, here, and then this is the pump which it can be used to automatically adding probes and wash probes. So you don't need to be there all night long. So the workflow for us to generate the spatial map for the retina is as follows. So we start out as a enucleated eye, and then we make a fresh frozen tissue, and we make a 10-micron section. So and from slide, and the slide is slightly fixed and hybridized to a probe of 386 in our case. That's selected from the single-cell RNA-seq for the cell markers. And then in parallel, we actually also add a few antibodies that highlight the cell boundary. So after the hybridization and staining, and then it goes through a multiple round of imaging to basically decode the probe, and so we can identify which transcripts are in the tissue. So here I show basically raw data. What you can see is the white spots are basically diabetes staining for the nuclei. As you can see, the retina of the mouse has 3 layers: outer nuclear layer, inner nuclear layer, and RGCs. So and then you can see a cloud of colors hovering around it. And then what happens is you can see, for the regions that basically on top of the nuclear region, you have more dense signal, while the layers between the 2 nuclear layers are much less. Those are the plex layer, which are basically synapse forming, and which makes sense to have less transcript. So if we look closely, we start looking at the marker probe, marker transcript expression. So what's shown here, this is the outer nuclear layer of the retina, and you can see the cells that are highlighted by the yellow lines. Those are cone cells, because we know that because we will see many probes that specific cones are highly expressed in there. You do see a few individual red dots. Those are likely to be basically background. So if we look at the probes against the raw markers, you can see all the rest of the cells are all the raw cells, and then the cone cells, in fact, precisely at the top layer of the retina, which is exactly where, based on previous results, is consistent with that result. Similarly, we'll look at the bipolar cells, which are interneurons connect the photoreceptor cells to RGC layers. As you can see, this is pan-BC marker, bipolar cell markers, highlight the top half of the nuclear layer. And this is a zoom in. And then if you overlay that with a subtype of bipolar cells like on, off, and RBCs, you can see, unlike the yellow dots, the other color dots have a specific pattern that basically highlights different subtypes. So what the data looks like. So we first look at how robust is the technology. So we did 2 biological replicate 1 and 2, and we just look at the value, the transcript value across the entire slide. So you can see they're highly reproducible with R-square of 0.94. And then we're also looking at, whether this robust results, the MERFISH results, consistent with the from the RNA-seq data. So we take this same retina and we did RNA sequencing, and we compare the MERFISH count. As you can see, where nicely positive correlated concordance are between these 2 technologies. So of course, now with the transcript, identified in the tissue, to the next step is to assign the transcript to the cell. So to do that, we need to first segment the cell and then identify the cell boundary. So as you can see in the left panel, is basically the white lines are cell boundary antibody staining. As you can see, compared to many other type of tissues, the retinal tissue, the cells are really, really packed, and then they are irregular shape, and then making it really hard to do segmentation. So by testing a variety of methods, and this is one of the segmentation results we got. So you can see most of the cells can be successfully segmented. So after segmentation, then now we can assign the individual transcript to the cell, and then basically obtain the expression profile for each cell, much like the cell gene matrix that we can get from the single-cell RNA-seq. To use that method, we can then to see, can we identify the cell major types in the retina? So as you can see from the UMAP, indeed, based on the MERSCOPE transcript profile, we are able to identify all the cell types we know in the retina, including some rare ones such as horizontal cells and RGCs. So this is basically now put back the cell label back to the tissue. As you can see, we see the outer nuclear layer have primarily rods, and then some cones on the top of the layer, and we can see the top half bipolar cells, as I mentioned, and then they are sprinkled with the horizontal cells, and then there's mural cells in the middle, at the bottom are the amocrine cells. Then you have RGC at the RGC layer. So that's consistent with what we know about the retina. So now our question is, okay, now we can find all the major cell types. Can we find the rare cell types, basically subtypes? So since the MERSCOPE itself only has 368 probes, the resolution is not very high. So therefore, we actually integrate the MERSCOPE data with the single-cell RNA-seq data to basically take advantage of the high resolution the single-cell RNA-seq can offer, because we profile large number of cells, as I mentioned, the quarter millions, and then to see whether we can then leverage this data to separate, to identify different subtypes in the MERSCOPE. So indeed, as you can see, we can successfully embed these two data together, and we can find all the bipolar cell types in MERSCOPE data. So not only we can find them, but also when we look at the proportion of different subtypes, the blue ones are the cell type proportion based on the RNA-seq, and then the orange ones based on MERSCOPE. Overall, they are quite consistent. So now we can then place all the 15 different bipolar cell types back to the retina, as you can see, very nicely laid out. So with this data, then we can explore some spatial relationship among them. So it has been known, or people in the field, that all the bipolar cell types are distributed randomly across. So when we now with the data and look at it systematically, in fact, I think we find it overall it is indeed quite uniformly or randomly distributed. However, there are some subtypes, for example, BC1A, 1B, which on the transcriptional level is very similar. They actually closely locate to each other, and they often form doublets because they basically near to themselves. So that's in this panel. You can see the green and red dots. They typically form a doublet or next to each other. Well, the other, most of the times, they are from really random distribution. And then we can see whether we can even have higher resolution now by combining the MERSCOPE with the single-cell RNA-seq data for the amaurosis cells. The amaurosis cells is much more complex, where they have more than 60 different subtypes. And then you can see when we combine the single-cell RNA-seq and with MERSCOPE, we can identify most of them, only a few clusters that cannot be distinguished. So then we start looking at their spatial relationship among the different amaurosis cell subtypes. Most of them are sort of random. There's no particular pattern, but there are some clusters form nicely. For example, this particular cluster at the left right corner is co-localized with the RGC cells. So when we look at it more closely, and what we find is there's 5 different amaurosis subtypes are localized closely to RGCs. So RGC itself form a layer at the RGC layer, and it's a single cell layer, and it has been known that some of the amaurosis cell, for a reason we're not so sure about, localized from the inner nuclear layer to the RGC layer, and those are called what we call displaced amaurosis cell types. As we know that starburst amaurosis cell is the one that's displaced, but based on the MERSCOPE data, we do find those it's AC17, corresponding to starburst, but the other 4, interestingly, has not been known to be displaced, and then they are all GABAergic ACs. So their function significance is still need to be investigated. We're in the process of doing that. So finally, we are thinking, for the wild type, MERSCOPE works really, really robust. Then we are wondering whether it is possible to detect some subtle differences in the mutant situation, for example, and use MERSCOPE. So we turn to this LHX3 mutant retina. LHX3 is a transcription factor specifically expressed in the bipolar cells. specifically in subset of bipolar cells, for example, BC1B and BC6. So in our probe, we did include this as a probe. As you can see in the wild type, the expression is in the nuclear layer where the bipolar cells are, and in the knockout mouse, which is the conditional knockout derived by 6-3 Cray, as you can see, the LHX3 expression is largely gone. You probably have maybe 5 to 10% left. So then we compared the morphology results obtained from the LHX3 knockout retina to the wild type. Overall, they are very consistent. All the markers genes essentially didn't change, except the LHX3 is dramatically reduced. So then we look at closely at the bipolar cell types. What we find is there's two subtypes, BC6 and BC1B, as I mentioned, where LHX3 is expressed, is dramatically reduced. This is the quantification. You can see 1B is largely reduced, and BC6 type is largely reduced. So therefore, from the morphology method, we can definitely pick up those rare cell types, the change in the rare cell types. So to summarize, morphology in our hand seems very robust and reliable in detecting transcript in the retinal tissue. So vast majority of cell types can be identified by leverage morphology with single-cell RNA-seq data. The spatial single-cell data provide interesting insights for the biology. Today we do not have time to present all of them, just show you a few examples. And morphology is really sensitive in detecting subtle changes in rare cell types. So for our lab, we are part of the HCA project, try to produce a spatially resolved single transcriptome and epigenome atlas for human eye across gender, age, and ethnic groups. We are currently incorporating morphology as a major technology for that project. So we also plan to apply morphology to characterize mouse and human visual system, including other parts of the eye other than the retina. For example, anterior segment, posterior segment. We are in the process of developing probes and testing it for it. And finally, we think there's a lot of opportunity to apply this technology to understand development biology, the mutant, and disease tissues. So I would like to end the talk by thanking for people who directly contributed to the project. Johnny, Selma, Jin Li, and then Qingnan are student postdoc in the lab who did most of the work. And then this is in collaboration with Jeff, and then Jason, Jingzhang, Ken, and Ling Gan. And then this is not possible without the collaboration with Rui Chen, especially one side to George and John for the help. Our work is supported by NIH, Retinal Research Foundation, and the Chan Zuckerberg Initiative. With that, thank you very much for your attention, and happy to answer questions.
Thank you, Dr. Chen, for your informative presentation. Dr. Zheng He, Director of Scientific Affairs for VizGen, will now join us for the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, why did you choose VizGen's MERSCOPE platform over building your own MERFISH instrument?
So, thanks for the question. So basically, as I talked in the talk, so our primary goal for this project is to identify all the cell types in the retina, and then put there and also provide the spatial information. So since the number of cell types in the retina is quite large, and the proportion of them are quite uneven, so we are really looking for something quite sensitive. So by comparing the variety of technology, we think the in situ-based method are way more sensitive than the sequencing-based method, which is limited by the enzymatic efficiency. So that's the primary reason. And the second reason is since the number of cells are large, so we do need a large panel. So most of the current spatial map method, the in situ-based method, the number of probes supported are quite low, like maybe 100 or 200. And MERFISH can definitely support to the 500 we have tried even a panel of 1,000. It works really well. So that's the second major reason. And then the third one is we are looking for true single cell technology because as you see, the cell subtypes are mixed together, and then they are so we do need true single cell instead of trying to use deconvolution method to identify them, which will be very difficult. So so I think because of the three reasons, we choose MERFISH as our first choice.
Thank you.
Thank you.
Next question. How do you interpret the heat map of spatial distribution of AC map on the slide with RGC labels? The heat map represents the abundance of the cell subtypes or their physical distribution.
Yeah, so thanks for the question. The heat map represents how closely the two subtypes are next to each other compared to the by chance. So the more red they are, meaning they more tend to get together. So in the RGC box, so those are basically identify the American cell types that locate close to RGC types.
Okay. Next question. Who or what applications would benefit from the insights provided by your technology?
Said who and what applications. So I think there's really a lot of possibilities opened up by this technology, as John already alluded to. So for the start, when you try to make a reference map, right, with the single cell RNA-seq, you find all the cell types and their transcriptome profile. However, you don't know how they located within the tissue. So so in the retina, for example, we are really interested in how the cell types are located, and then also build on top of it, then we can potentially think about neural circuit connectivity. So we do want to know their spatial relationship. So that's the first, that's just reference. But second, I think many applications, for example, the cancer situation where you want to understand the within tumor or at the boundary, what happened there, in the retina situation. For example, if you have retina degeneration disease such as AMD, there's a juice and deposit. People be really interested in understand the cells next to the lesion versus the cell far away from the lesion, what their behavior is. So I think this just really a long list of things that the people can gain insight by having the spatial information on top of the single cell transcriptome you can get.
Thank you. Next question. Have you found endothelial cells or astrocytes or microglial cells?
So currently in our panel, we didn't put those marker in there. Those cells are really, really rare. I think if we put marker there, we probably can pick up them because we do pick up extremely rare cells. But we haven't really tried in this particular case.
All right, thank you. Next question. How long does a single experiment take, assuming usage of the entire 1 cc imaginable area, including both the imaging and hybridization washing steps that are performed on the machine?
Yeah, so currently our protocol, it takes about 4 to 5 days from the time you're section the tissue, start hybridization, and clearing and hybridization to the image.
All right, thank you. I'm sorry, continue.
Yeah, right. I just want to say, it really takes 4 or 5 days, but you don't need to, the hands-on time is much less than that. Some days just few hours, some days very fast.
Yeah, perhaps this is Jiang speaking. Perhaps I can also add a further note on what Dr. Chen was saying about the imaging time. For MERFISH and MERSCOPE imaging, it's often divided into 2 phases. The first phase is sort of off the instrument, sample prep from the histology cutting to hybridization and some of the washing steps. That usually takes a couple of days to prepare the sample. And then there is a second step, which is the imaging acquisition. Again, for imaging 1 square centimeter tissue, about 500 gene panel, that usually takes about a day or so.
Okay, great, thank you. Next question. Is is there a platform to be able to locate expression of vast variety of different link RNAs?
We haven't tried it, but there's no reason why not. So as long as the link RNA sequence is unique enough, you can program, then you should be able to see that. Yes.
Yeah, I can also add on a little bit, because actually link RNA and non-coding RNA has been profiled by Murphy's technology in the past. So there has been demonstration of Murphy's imaging more than 10,000 genes in cells, and in that research paper, actually non-coding RNAs were actually included, and they were able to identify quite a number of those non-coding RNAs enriched in different organelles or nucleus.
Okay, thank you. Next question, does PFA fixed tissue sample work similar as fresh frozen sample? And if not, what are the major differences?
Yeah, this is probably a good question for John, because he's more experienced in this.
Yeah, thanks, Dr. Chen. So yes, so the Murphy's protocol works for both fresh frozen and PFA fixed frozen tissue types, and there are I think two key differences. The first difference is that for PFA fixed sample, there's no need to fix the tissue slice anymore. After cutting, you can directly go for permeabilization. There's no need for permeabilizing that anymore. The second key difference is in terms of the background removal at later step. So for Murphy's protocol, the sample prep procedure after hybridization and washing, we do have an autofluorescence removing step to remove the proteins and lipids, and fixed frozen tissue, because they are more cross-linked, they use a harsher removal procedure at an elevated temperature. And in our hands, we have actually demonstrated across many different mouse tissues that it works very similarly for fresh frozen versus fixed frozen sample types.
Thank you. Next question. Regarding the retinal preparation, have you tried flat mount or only cross-section?
So far, we only tried cross-section. We actually tried once with the flat mount. I think there's a lot of challenges there because the imaging, because also how do you, when you try to prepare the slide, to move the tissue over. So, but I think based on our, one-time try, we think it's possible, but definitely some optimization is needed to do that.
Okay, thank you. Next question. Are you planning to study ciliopathies that affect the eye, in particular Bardet-Biedl syndrome?
So yes, we are. As we showed in the presentation, we tried on the mutant mouse. So we definitely were very interested to look at human disease eyes, for example. But it just the sample availability is really limited. So, but I think we really interesting study to follow up.
Thank you. Next question. How critical are antibodies to define cell boundaries?
So the cell boundary antibody, conjugate antibody, is actually we receive directly from Rui-Chen, and they have 3 different types, and then we use all of them at the same time. So I think in our case, the second one, the channel 2, works the best, but not for all the layers. So I do think depends on the tissue. Maybe different type will work better, or sometimes you probably need to use a combination.
Okay, thank you. Next question. Could you delve deeper into how MERFISH allows for more robust readout than the typical in situ hybridization method?
Yeah, this is actually a question for John, but based on my understanding, I can start. It's because you have 30, I think 30 to 50 different probes are used for each transcript. So the readout is much more robust because as long as some of them work, you will see it. The second one, they use the robust method, so they can also do a lot of error correction, so we'll have much less false positive. So based on the published paper, their sensitivity is at like 80%. It's much higher than the single-cell RNA-seq, which is around 20%. So John, do you want to add more?
Sure. Yeah. Thanks, Dr. Chen. You hit the point very well already. I just wanted to further extend to mention that MERFISH is actually built up on the single molecule FISH, which is in situ hybridization method already. However, we do have something that's called error-robust barcoding. And what that really adds is that because if you are to sort of do single molecule FISH imaging, there's chances that the probes may sort of due to unspecific binding that will be false positives or false negatives. However, by using this binary barcoding scheme and assigning each RNA transcript with a defined barcode, then even if there is some error due to the unspecific binding or loss of signal, then there is ways for us to actually detect the error and correct, and that actually makes the measurement very, very accurate. And another thing that I think sort of speaks for the sensitivity and also the power of it is that this actually also makes it highly quantitative, that we are able to really quantify the gene expression even for very low expressed genes. And these two features doesn't really compromise, as Dr. Chen was mentioning, it doesn't really compromise the detection efficiency. We still preserve that very high detection efficiency of single molecule FISH because there are so many probes used for a single gene.
Great, thank you. Next question. For what type of projects would you recommend MERSCOPE, considering this is a targeted approach as of today?
So I think, the MERFISH, of course, is limited. The major limitation of the MERFISH right now is you need to pre-design a set of probes. I think it's probably quite reasonable to think, up to 500 or maybe 1,000 genes will be reasonable or relatively easy to implement or optimize. So therefore, if you're looking at a sort of discovery experiment where you really don't know, you want to do unbiased discovery, then this will be some limitation. It is possible to, you already published, you can look at 10,000, transcripts, for example. But I think those are relatively, challenging basically right now. Based on my experience. So I think, if it's truly discovery, this will be a little bit harder. But with that said, I think by combining with the single-cell RNA-seq with MERFISH, you still, and with some computational analysis, you still can basically answer that question, even if it's for discovery.
Okay, thank you. Next question. Why do you think the correlation of MERFISH with bulk RNA sequencing was only about 0.83 and not higher? What is the RNA sequence expression measurement used here? kpkm per Q, exon count, or counts on the region of MERFISH probe?
Yeah, so the RNA-seq is the bulk, RNA-seq. It's a full transcript RNA-seq we happen to have done on the mouse retina. So it was done not exactly, the same sample as MERFISH was conducting on. So I think part of the reasons could be from here. And the second one is the chemistry, right? So the RNA-seq is basically you go through CDA and then RT process, and then you're counting the transcript per million, and then from the MERFISH it's essentially the count, how many spots you see that transcript. So I think this the main reason is probably in the chemistry. One is really just hybridization based, the other is sequence based. There's some systematic bias, but overall they are quite similar.
Okay, thank you. Next question. There are not many cells in trabecular meshwork. Is it possible to use MERSCOPE platform?
It's really tiny, tiny structure, yes. But I think since MERFISH is a single cell resolution, so if we do the section and then contain the trabecular meshwork, I don't think there's any reason not be able to do it. I think the answer is yes, we should be able to do it.
Great. Next question. How long do transcripts have to be to allow using MERFISH?
So most of the time the reason you cannot include a transcript in the probe set is because the transcript may be very, very highly expressed in your tissue. And then because of the optical crowding, those are excluded, or the sequences are very similar, like duplicate genes. But I don't think the length will be a limited factor, because although it's common to use 30 to 50 probes, but it's not necessary to have that many. It is possible to have just a few probes. That's my understanding. John, do you want to add?
I can add a bit on that. So currently, the MERSCOPE probes are usually designed 30 to 50, and indeed, we sometimes even lower the amount of probes required for targeting each gene. So again, since each probe is 30 nucleotide long, so if it's 30 probes per gene, then sort of the minimum length, it will be around 900 nucleotides. And if you reduce the number of probes, then it will be a shorter transcripts. But there are strategies around to actually use fewer probes. For example, there has been some early demonstrations for MERSCOPE in the literature that actually using amplification, you basically can use much fewer probes to get around 400 or even short 400 nucleotide long genes. And with that, then the majority of the genes probably will be targetable with the amplification strategy. So these are some of the things that Wisdom as a team is still working on to sort of roll out as a product, but proof of principle wise, it has already been well demonstrated in the literature from our technology.
Great, thank you. Next question. Would you recommend the cell boundary detection kit in less dense tissue as well?
Yes. Sorry, is it broken?
I can perhaps answer this because this is more related to Wisdom's cell boundary kit. So indeed, so less dense tissues, for example, the central nerve system, like the brain, the spinal cord, usually there's no need for cell boundary kit because the cells are very separate. The cell body needs are very separate from each other. So for many of those projects that we have seen, they don't need to use cell boundary staining kit. It is those tissue where cells are very tightly packed against each other. Then the benefit of including a cell boundary kit then really helps to define the shape of the cell, and then you get true single cell identification.
Okay, thank you. And it looks like we have time for one more question. How stringent about the platform for SNP or mini-indel detection on the transcripts? What is the dynamic range of signal detection in terms of GEA or genes with the same set?
So I think because the number of probe people you can use, you put down over the transcript is large, so I don't think it's really sensitive to like polymorphism. If that's some things you worry about. So I don't think the MERSCOPE have trouble to pick up those. In terms of the dynamic range, I think it's basically the lower sensitivity, the low end ones, you can identify one transcript in the cell, right? So it's very sensitive, and then the high end is just subject to the optical crowding. So typically in our hand, we definitely can see a dynamic range of 10 to the 4th. That's not a problem.
All right, thank you. And thank you again, Dr. Chen and Dr. He, for your time today and your important research. We would also like to thank LabRoots and our sponsor, VizGen, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.